thank you. I'm Elizabeth Jaramillo. I'm a postdoc at the Jet Propulsion Lab. And today I'm going to be talking about a method I've been developing for the analysis of organic and inorganic ions using capillary electrophoresis with capacitively coupled contactless conductivity detection, or CE-C4D for short. So the target for this method is really analysis of the samples from this ocean world. I don't have to explain to this group why these worlds are important or interesting, so I'm not going to waste my time doing that. But I'm going to say that one of the things I would like to possibly use this for is incorporation into a potential larger mission for life detection. And there was this wonderful paper that was published that sort of delineates how you might search for life and detect life on other worlds. And the method that I'm developing would sort of fall on the rung that describes molecules and structures conferring function. So I would be looking for patterns within classes of molecules that are not random and therefore might be indicative of life. And the classes of molecules that I'd be looking at are things like amino acids or carboxylic acids, because these are things that we find for carboxylic acids. They're important intermediates in metabolic processes, and they can sort of be components of cell membranes and things like that. I'm also interested in this method, and it probably has its strength mainly in habitability assessment, and I want to take a moment to make sure that we are clear about the difference between life detection and habitability. So my Cliff Notes version of this is for life detection, you're looking for evidence of some sort of metabolism. Habitability is just asking whether or not that environment could support a metabolism. And it's much more complicated than that, but those are the Cliff Notes for now. And so the basis for looking at a habitable environment would be looking for something like water, organic molecules, and energy sources. And having these are not simply enough. You need to have them sort of co-located so any organism that might be present could actually use them for metabolism. So to do this, we're going to first search for these organic molecules and energy sources in the soluble chemistry of the environment. And that will ensure not only that they are co-located, but also that you have a solvent that is able to participate and support the electron transfer processes that are essential for metabolism. So there are different organic molecules that we will be looking for and different energy sources that we will be looking for. The specific targets that I've sort of used to develop my method, for inorganic ions, we have over here, we based it off of seawater for a lot of them because if we're looking at ocean worlds, our ocean is sort of a decent model for it. Also, we suspect there will be a lot of chloride, there will be sulfate. So we're looking for bromide, chloride, carbonate, sulfate, and we're also looking for metabolic energy sources. So microbial life on Earth can use oxyanion species like sulfate and nitrate and perchlorate in order to metabolize. I also want to take a moment to specifically focus on perchlorate just briefly. If we are expecting these ocean worlds to have a lot of chloride, and we think they might be highly oxidizing, I think it is important that we consider the presence of perchlorate as a potential ion that could either interfere or support metabolic life in these oceans. And so I wanted to make sure that that is part of our search in these inorganic sources. We're also looking for carboxylic acids. So I sort of split them into these short chain carboxylic acids and potentially longer chain ones. So here we have a sort of loose selection of ones that are found in meteorites that are shorter chain. They're monocarboxylic acids, they're branched, they have hydroxy groups attached to them. And then we have these, which are sort of di and tricarboxylic acids, which are products and intermediates in metabolism. We're also interested in some monocarboxylic acids that are longer chain lengths that are derived from lipids and could be potential components of cell walls. So the technique we're using, CE, is ideal for this sort of analysis because it inherently separates charged species in an aqueous solution, which is exactly what we're looking for. So to do a CE experiment, you have a thin capillary, about 50 microns in our case, and we fill it with a background electrolyte. We inject sample into one end, and then we apply a voltage. And what happens is, under the effect of that voltage, the charged species in the sample will separate based on their mass and their size and their charge. And so at the other end, you can place a detector, and you can detect the different species that are already separated. The detection method that we have chosen is a form of conductivity detection, and we chose this because it will detect any charged species. So we don't actually need to know what the charged species is, so long as we can detect that it is different from our background electrolyte. It doesn't require any labeling or derivatization, so we can use this in conjunction with other detection methods. So we could put perhaps the C4D detector in line with a mass spec and get even more information out of it in the end. see, right. So if you noticed when I talked about the targets that I was interested in, I talked mostly about anions. And the reason for that is that I have a lot of lab mates who have done a lot of work on the cation side of this. Here we have a paper that was published last year by a fellow postdoc, Mauro Ferreira Santos, and I encourage you, if you're interested, to look up that paper. And what he did was he used the same CE-C4D technology in order to separate inorganic cations and amino acids simultaneously. And the background electrolyte he used for that was acetic acid. And then we have another method that was developed by Jessica Creamer, who's giving a talk later today that I encourage you all to go to, that has done, she's done excellent work developing chiral separations of amino acids with very low detection limits. And the electrolyte that she's been using has been based on sodium tetraborate. So as I go forward to develop my method, I'm looking for using it in these in situ environments where we're really trying to minimize the complexity of our instruments and our methods because we don't have a lot of space, we don't have a lot of power. And so we want to have it be as simple as possible, but we're not really willing to give up any of the science that we're looking for. And so my approach to doing this is to take those cation methods that my lab mates have developed and develop an anion version that is compatible. So it should be run on the same capillary with the same equipment, and ideally use as many of the same reagents as possible. So this is exactly what I did. So method one, I basically took the acetic acid-based method that was developed for the simultaneous separation of inorganic cations and amino acids, and I titrated it with sodium tetraborate until it reached around the pKa of acetic acid, and I used that to separate, whoops, that's the wrong button. I used that to separate the inorganic ions that I was interested in, and then the small, very highly charged oxalic acid. And then I basically did the inverse in order to develop the carboxylic acid version of this. So I took the sodium tetraborate that's being used for the chiral amino acids, and I titrated that with acetic acid to about the pKa of sodium tetraborate, and I used that to separate out these carboxylic acids. And this is very interesting because we can get, it's very convenient, I don't know if it's interesting, because we have this division here, and at this point, everything below that are these sort of monocarboxylic acids, these hydroxy acids, these things which are typically found in meteorites and the like. And on the other side, we have the highly charged di and tricarboxylic acids and things that are typically found as metabolic intermediates and might be more interesting in that respect. We were also interested in these longer chain carboxylic acids. So these are the potential components of cell membranes and things like that. And so we find that earlier on, even earlier than many of the hydroxy and branched monocarboxylic acids, we get this suite of them that come out. I ran this between a 3-carbon long monocarboxylic acid all the way up to a 20, and we were able to get up through 14 carbon long monocarboxylic acids, which starts to approach your solubility limit for these. So I think this is pretty indicative that if we're looking in the soluble chemistry, we can get most of them using this method, and we can get them in chunks along the way. So we also felt it was important to test this in a natural sample because standards are great because standards are great. So I found a pond that was certainly full of life. It had a lot of green in it and a lot of turtles. And so we analyzed the sample from there. We did not dilute it. The only pre-treatment that we did was we hydrolyzed the cells by heating them up to 180 degrees for about 30 minutes, which would hopefully break apart the membranes and get anything that was inside of them out. And when we ran that, we found the chloride and the sulfate, which are the dominant anions, inorganic anions in that solution. And then we also found citric acid, which we would expect because if you have a lot of metabolism in a very large life-filled pool, you would expect citric acid from the citric acid cycle to be there. We found carbonate, and we found a tiny bit of monocarboxylic acids. And then we found this little peak down here, which turns out to be indicative of potentially a lot of amino acids. So we can't separate any amino acids with this method, but what we do get when we spike a solution with amino acids is this immediate large dip that looks exactly like this, which may be indicative of the amino acids. Great, so I am ahead of time. So moving forward on this method, there are things we want to do. So right now we've tested in this very friendly, life-filled pool, but we want to be able to make sure that we can use this on things that are a little more analogous to the ocean worlds that we're hoping to use it on in the future. So we did some field work, and we went and collected samples from Owens Lake and Mono Lake. which are two hypersaline alkaline lake environments, which could be analogous to these. And so we're hoping to use this method to analyze those samples. And then we're also beginning to develop a simultaneous method, because while it's beautiful and simple to say we're just going to use the reagents from these cation methods and use the capillary and use all the equipment, and there's a simplicity that makes that really exciting, it still requires two analyses. And so if time is your issue, then it's actually not as simple as we had hoped. And so I'm attempting to develop a new method that will do this all at once, and we're still using the same acetic acid base, but we're adding triethylamine instead of borate to adjust the pH of this. And using this, we can separate all of the primary inorganic ions with the exception of the chlorate and perchlorate. So both oxychlorine species seem to coelute. And we can actually get all of the carboxylic acids that we were interested in all the way up to C10. So it kind of crashes out at that point. But we're able to get all of the other ones, including citric acid, and we're able to detect carbonate and things like that. So with that, I would like to have a shameless plug for the rest of my lab mates. So we're part of the Chemical Analysis and Life Detection group at JPL, and a lot of us are giving talks and posters this week. So this is Nate and Constantine have posters tonight, and they're talking about developing the hardware and qualifying the hardware for the instruments that might use these kinds of methods. They've done a lot of terrific work, so I suggest you go by and talk to them. I already talked about Jess's talk for chiral amino acids. Fernanda is giving a talk on her chemical laptop, which is a portable microchip device. It's automated, and she's done a lot of excellent work with that, so you can see that tomorrow. And Florian is giving a talk about his liquid extractor. So even if we had a soil sample or something that wasn't necessarily already in our perfect liquid form, he's developed a tool that will help us get it into the form we need for this. And then on Thursday, Peter, our group supervisor, is going to give an excellent overview of all of the work that we've been doing developing these types of techniques for these environments. And then Aaron is going to be talking later about a microfluidic ion analyzer that we've been developing as well, which is complementary with all of this. So with that, I will take any questions.
We've got plenty of time for questions.
Hi, Lauren Seiler from HUI. So there's a pretty big difference between a small amino acid like glycine and like a really huge one like proline or tryptophan. Do you have any idea why this technique can't tell the difference between those things?
Yeah, so it has to do with basically the speed with which they move. So the method that detects them but doesn't separate them is because well, actually I'm not sure it would even measure glycine. They all just move so fast under that, because in addition to separating based on charge and size, when you apply the electric field, you also get what's called an electroosmotic flow. So the solution, the background electrolyte, gets pulled along with these ions toward the negative end. And so if that's happening, if it's too fast, it can sometimes squish together those small, highly charged positive ions. And so the reason the negative ones separate is because they have a draw backwards that the positive ones don't have. So you really just get them all at once. And you don't get any separation because they move just way too quickly.
Got it. Thank you.
I have a question. I guess it's easier for me jumping from here. So when you mentioned the pond with the turtle, I would expect a lot of life there in terms of abundance. But from the peak, just the way they look like, they look very, very kind of like small and dry.
Yes.
Could you repeat the question into the microphone?
Okay, yeah, so she was asking about the size of the peaks given that the abundance of life we'd expect in that pond, right?
Yes. So
Okay.
it's more qualitative
Yes.
rather than quantitative.
Well, so it can be quantitative. I didn't quantify that because I'm still working on really making that a little more, I guess robust would be the phrase. But especially with the monocarboxylic acids, probably what's present there just isn't soluble in the water. They're probably still longer chains that we just can't get to. In terms of the citric acid, I'm not sure how much I would expect to see, to be honest. I haven't quite finished doing that. It was our sort of first attempt at an analysis like this. So it could just be that when you break apart a cell, right, maybe, I'm not sure how much citric acid you expect to get out with that metabolism. But it's something that we're kind of looking at and trying to figure out how much we expect sort of per cell and how many cells we might need to say that we found something, and iteratively like that, yeah.
Okay. So let's thank our speaker again, Elizabeth Jaramillo. And